AC కూడా ఆన్ చేయి. Yeah, children, good morning. Today we are going to start with a chapter called Plant Growth and Development, and we need to do the last rites of this chapter as well. The time allotted for us will be half an hour, and this is the maximum time with which we can complete this. Without wasting a single minute, we'll start with plant growth and development. The first point here, we'll talk about the characters of growth. What did you study in the living world? Growth is not a defining feature because it is observed even in non-living. 2. Growth is absent in dead. 3. Growth in plants is indeterminate thanks to apical meristems. 4. Growth in animals is determinate. After some time, the growth is going to cease or stop. 5. Growth is external in non-living things. Growth is internal in living things, intrinsic. And the two twin characters of growth is increase in mass and number. So these are the points which you studied in the first year part in living world. So we'll continue from there. The true definition of growth is increase in protoplasm. When there will be increase in protoplasm, children, this is simple common sense. There should be more anabolic changes when compared to catabolic. That is the reason why there is an increase in protoplasm. So it means that growth occurs when anabolism is more when compared to catabolism. Is anabolism there in growth? Yes. Interestingly, even catabolism is also a part of growth. So catabolism also have a role in growth, because only when anabolism is more than catabolism, you see growth taking place. Children, breakdown of glucose is needed. It's a catabolic process, releasing energy, and that energy is responsible for cell division. All of you know that. Young leaf is going to grow, we say that. But after some time, what happens is the leaf become old. When the leaf become old and mature, it's going to shrivel, it's going to wilt. There, the catabolism is more when compared to anabolism. Then opposite to growth take place, and that is called degrowth. That leads to senescence or aging of plant parts. A very important point is that anabolic rate is more when compared to catabolism when growth is occurring in plant part, is a point all of you should remember. Next important point. Growth is any change which is irreversible or permanent. Understand that. Any change which is irreversible or permanent is said to be growth. Now, all of you heard that a stromatal complex contains guard cells. During daytime, the guard cells become turgid. By night time, the water moves out and become flaccid. Can we call that guard cells are growing during daytime? No, because this change is only temporary. A wooden box or wooden door in rainy season is going to swell by absorbing water, and in the summer season, they become normal. Can we call it as a growth? No. Why? Because there the growth is temporary. It is not permanent. So understand, any irreversible change is said to be growth. Now, what happens in, before I start talking about the remaining parts of growth, the growth in plants and all that, the first page of your textbook talks about every organism starts its life from zygote. 2. First, a seed produces seedling, seedling produces a plantlet, and plantlet produces a mature plant. Question you're going to ask me is, what is the difference between seedling and plantlet? Seedling is a young plant which is yet to photosynthesize. Plantlet is a young plant which is capable of performing photosynthesis by its own. Then there is a diagram in your textbook, epigeal germination. The cotyledons are brought above the surface of the soil because the hypocotyl, because the hypocotyl elongate. That is observed in Dolichos lab bean. You remember that example? Next important point, during growth, what happens first? Vegetative organs precede reproductive organ development. Why like that? Because only when vegetative organs develop, photosynthesis take place, energy is produced, food is produced, which can be used for growth of reproductive organs. Then, which organs are going to show indeterminate growth? Root and stem. Which organ shows determinate growth? Leaves, flowers and fruits. Which organs does not show any periodic fall? Surely, root and stem. Which organ shows periodic fall? Leaves, flowers and fruits. Even this point is also important. And the last point, what is the meaning of development? All the changes occurring from seed germination to senescence, aging of plant parts, all that put together is said to be development of an organism. If I talk about a plant. All right, does it include germination of seed? Yes. Vegetative growth? Yes. Reproductive growth? Yes. Differentiation? Yes. Dedifferentiation? Yes. Dormancy? Yes. Senescence, aging? Yes. Don't think that development include only constructive changes. So there are some changes which are dormant changes also take part, is also a part of development. You also remember that very important point. Now I come back to the growth in plants, children, because this chapter restricts about the growth in plants only. Growth in plants, there are two types of growth, primary growth and secondary growth. Let me tell you the difference. Occurring in all plants, primary growth. Occurring only in dicots and gymnosperms, secondary growth. Increase in the length of the plant organ, primary growth. Increase in the girth or circumference of the plant organ, secondary growth. Apical meristems and intercalary meristems contribute in primary growth. Lateral meristems like vascular cambium and cork cambium contribute in secondary growth. Primary growth cause increase in the length, whereas secondary growth cause increase in the diameter. So these are the points which you have to remember between the primary growth and secondary growth in plants. Don't make any errors with regard to this topic. It is linked a little bit to anatomy. You should also remember lateral meristems help in secondary growth, vascular cambium and cork cambium, apical intercalary meristems help in primary growth. That completes the growth in plants. The next important one. What is the absolute definition of growth? We already discussed that. The absolute definition of growth is increase in protoplasmic content. Your textbook says which is difficult to measure. True. Increase in protoplasm cannot be measured. It can be equated indirectly with increase in dry weight, increase in fresh weight, length, surface area, weight, etc., etc. Your textbook talks about what are the parameters which can quantify, which can be indirect measurement of growth. Example, maize root tip. The increase in the number of cells there represent growth. All right, watermelon. Increase in the cell volume. All right, weight. That is said to be growth. Pollen grain producing pollen tube. Increase in the length of the pollen tube is a measurement for growth. Leaf. Increase in surface area is a measurement for growth. So, simple point. Growth cannot be measured properly because increase in protoplasm cannot be measured. Therefore, there are other parameters which can be equivalent to increase in protoplasm as we have discussed here. The next important point what we have is the phases of growth. What are the three important phases of growth? In first year morphology you studied. All right, the part of the plant which is far away from root stem junction. This is root stem junction. Far away from root stem junction, English word far away, distal. So this is the distal most part and this is the proximal. This is the parameter. From here it is far away. Therefore, this is distal. This is called root apex. And this is called elongation. And this is called maturation. This is C is maturation. This is elongation. This is root apex. And this is maturation. When we talk about these regions, by talking about these regions, I can even talk about the phases of growth. Root apex. I'll tell you, the root apex contain meristematic cells, apical meristems. What are the characters of apical meristems? Listen carefully. 1, they have thin cell wall, no secondary wall. 2, they have dense cytoplasm. 3, they contain conspicuous nucleus. 4, they contain plenty of plasmodesmata because they are active. They need to divide, so therefore cell-to-cell connections are more. 5, vacuoles are either not developed or even if they are developed, they are too small. So these are the characters of cells present in the root apex. So the cell division is the first phase of growth with regard to root. The second phase of growth is elongation. A parallel line diagram, if you draw, there is more diameter here when compared to here. So it's very clear that elongation is the major contributor of growth in root. How can the cells elongate? Is it absorbing air just like a balloon? No. Cells elongate by absorbing more amount of water. Where is this water stored? Inside the vacuole. So it means that these cells should have a large vacuole. By this time, the cell wall is reasonably well developed. You see the primary cell wall, all right, properly developed here. Protoplasm is pushed towards periphery because of large central vacuole. Vacuole will be storing plenty of water. This is the significance of elongation zone. Then we talk about the maturation zone or maturation phase. The cells lose the power of division, differentiate, and if they want to perform a proper function, they have to modify themselves permanently. Your textbook talks about lot of maximum protoplasmic modifications occur in the region of maturation. The cell wall becomes relatively thick, maximum protoplasmic modifications in order to suit the function which they want to perform. That is the characteristic of the last phase of growth. That is said to be maturation. We don't have time to repeat again. All right, that's about the next important unit. That is about the phases of growth. Then we talk about the next one. What we have to discuss is about the two types of growth rate: arithmetic growth rate and geometric growth rate. Now, what do you see in arithmetic growth rate? A cell divides into two. One cell is going to become permanent. The other cell divide into two, and one cell become permanent. The other cell divide into two, and one cell become permanent. Every time out of two cells, only one cell divide, the other cell become permanent. What did you study in mathematics in your lower classes? Arithmetic proportion. This is what your mathematics teacher said. 1, 2, 3, 4. It's more or less a uniform. So if I show you the graph, the growth, all right, this is L0. All right, length at the initial. After some time, there is a growth. So naturally, this will be the LT. All right, with regard to length, and this is the time. So what is the definition? After, with passage of time, there is an increase in the length of the plant organ. Your textbook talks about increase in the length of the plant organ like root and stem. That's a wonderful example for arithmetic growth. All right. Now, what did you observe after time t? LT is equal to L0 plus RT. All right. So it means that the length after time t is equivalent to the length, the initial length plus the relative, what is called RT, where R stands for what is called arithmetic growth rate, which is more or less constant. More or less constant. All right. And that is the wonderful example for this is increase in the length of the root, increase in the length of the stem is a wonderful example for arithmetic growth, because this number is arithmetic. That's what you know it. And this increase is also very much linear with passage of time. Therefore, this is the arithmetic growth, which is very simple to remember. Then we talk about the geometric growth. Now, what do you see in geometric growth? This is the cell dividing into two. Both the cells are capable of division. Look here, this is the difference. Both the cells are capable of division and they're dividing like this. So if you look about the graph here, then this will be how it looks. Initially, the number of cells are less. This is said to be lag phase. After some time, all the cells divide. And after some time, there will be plenty of cells. That is said to be exponential or log phase. And after that, the deficiency of nutrients is the reason why the growth is going to cease, and that is said to be stationary phase. So lag phase, log phase, and stationary. It looks like S-shaped curve. This is said to be a sigmoid curve. This is a wonderful example for geometric growth. Now, what are the examples for geometric growth? Your textbook talks about many examples. Example number 1. Majority of cells, tissues, and organs, say for example, leaf, flower, and fruit. What did you see, children? Leaf, early leaf, young leaf is small. It is comparable to lag phase because the growth rate is very less, because the number of cells in that is also less. Suddenly, there is a far greater increase in surface area because enough nutrients is available. So therefore, it's going to show an exponential phase called log. And after that, once the leaf is going to become senescent, all right, the growth is going to cease. That is says to be stationary phase. So flowers, fruits, leaves. So majority of the plant organs is going to show a sigmoid curve because their growth depends upon the conditions outside as well. Unlike relative growth rate where the growth is more or less constant. Here the growth is not constant, unlike relative growth rate. One more example for this geometric growth is a tree which is experiencing seasonal changes. That is also an example for geometric growth. You may ask me how. In early spring, the leaves, flowers, and fruits will be very, very small. It is comparable to lag phase. In the late spring, when the conditions are very much congenial, every part of the plant organ is going to grow. And that is said to be a log phase or exponential phase, where the growth rate is very much high. Come autumn season, all the leaves fall down. Then the growth rate is very much less or zero. So that is said to be a stationary phase. So you mean that, again, that is said to be an example for a sigmoid curve. That is a very important point to remember. That is about geometric growth. And the equation for geometric growth is Wt is equal to W0 to e power RT. And the most important one, this R stands for what is called the changing efficiency index, the capacity of the plant organ to grow depending on the circumstances around it. That is the reason why I call it as efficiency index, or I can even call it as geometric growth rate or efficiency index, because it all depends upon the circumstances or the situations in which the cell or organ is there. That is going to be a contributor for the growth of it. This is the equation for what is called geometric growth rate. I want you to remember this as well. We don't have much time more than this. Then the next important one. A smaller leaf is going to become 5, is going to become 10, the surface area, 5 centimeters square initially. Then it is going to become 10 centimeters square in surface area. Then this is the leaf which is 50 centimeters square in surface area, is going to become 55 after some time. What is absolute growth rate here? Final minus initial. That is 10 minus 5. So absolute growth rate is 5. Here also absolute growth rate is equal to 5. Final minus initial. Then what is the relative growth rate? Relative growth rate is equal to absolute growth rate. Absolute growth rate by initial into 100. So what is the relative growth rate here? Absolute growth rate is 5 by initial. Initial is also 5 into 100. So it is 100%. 100% is what is called relative growth rate of smaller leaf. Here same. All right, absolute growth rate is 5 by initial is 50 into 100. It is only 10%. It means that both these leaves, smaller and larger, have the same absolute growth rate, but the smaller leaf has got more relative growth rate when compared to larger leaf. This is an important point to remember. That is the max your examiner can ask you. We'll go without wasting any time. The next important one. What are the conditions for growth? The first important condition for growth is water. Water is needed for growth because in the presence of water, the cells become turgid and increase in size. Second important contributor for growth is oxygen. In the presence of oxygen, respiration occurs, energy is liberated, and that energy is responsible for cell division. Third important contributor is nutrients, because nutrients make the respiratory substrate. When these respiratory substrates are made up of nutrients, naturally when they get oxidized, energy is released, which is responsible for growth. Apart from these three, light also helps in growth. We'll be discussing that when we go to auxins. Gravity also contributes in growth. These are all the factors which contribute in growth. These are all extrinsic factors. There are some intrinsic factors which are intercellular, from moving from one place to another. Wonderful example for that is hormones. And there are some intrinsic factors which are intracellular. Wonderful example for that is genes. We'll come back to that concept a little later. Then we talk about the next important one, the concept of differentiation, de-differentiation and redifferentiation. Listen carefully, time and again you're making mistakes here. When a meristem is going to become permanent, then that process is called differentiation. Means when the meristem lose the power of cell division and become permanent. Your textbook says the differentiation is open. Yes, children. The meristem have an option of giving rise to a permanent tissue. And that permanent, different option, that permanent tissue based on location can perform at different functions. If I give an example for that. Imagine the shoot apex. It can give rise to leaf primordia and leaves. The same apical meristems in the axil of leaf can give rise to a branch. The same apical meristems, if you remove it and grow in a tissue culture medium, it can give rise to a complete plant. Means the ability of the plant organ to differentiate is open, is capable. That means it has a capacity to give rise to any type of permanent tissue which is capable depending upon the location. This process is called differentiation. Not only growth is open, even differentiation is open is a very important point to remember. And growth is localized in plants because the growth in plants is only at the tip, at the lateral meristems, lateral region, and also at the internodal region, nodal region, intercalary meristems. That point also you remember. When the permanent cell, remember the flashback, and become meristematic, then you call that process as de-differentiation. Sometimes the permanent cells, especially parenchyma, sometimes collenchyma, they remember the long back their meristems. So when they turn meristematic, then that process is called de-differentiation. So when such meristems, means secondary meristems you call, when such meristems lose the power of cell division and become permanent tissues, then it is called redifferentiation. When such secondary meristems lose the power of cell division again and become permanent tissue, then you call that process as redifferentiation. Chalo, I'm going to give you some examples for all three. Listen carefully. Now just concentrate what all I'm talking. Formation of epidermis, differentiation, because it's a permanent tissue forming from an apical meristem called protoderm. Cortex, hypodermis, general cortex, endodermis, differentiation. It arises from an apical meristem called ground meristem by differentiation. Then stele means pericycle, primary xylem, primary phloem, medulla. All right, it arises from procambium. Procambium lose the power of division. So therefore, all of them is differentiation. Epidermis, hypodermis, general cortex, endodermis, pericycle, primary xylem, primary phloem, medulla, medullary race, mesophyll, guard cells, subsidiary cells, trichomes. They're all examples of the products of differentiation because they're all permanent tissues seen in primary plant body. Then, interfascicular cambium. Some of the cells of medullary race de-differentiate and give rise to. So, interfascicular cambium and cock cambium are the wonderful examples of the products of de-differentiation. Even tumor formation, tumor, tissue culture, tumor are also examples for de-differentiation. Suddenly, the cells divide madly and give rise to and develop into swelling, tumor. That is de-differentiation. Tissue culture, from callus you can develop the entire plant. Suddenly, the cell which is non-dividing, take a leaf cell, non-dividing, you drop into the medium containing hormones, it starts dividing. So that's also an example for de-differentiation. Then, what are the examples for redifferentiation? I told you, secondary meristems giving rise to permanent tissues is redifferentiation. Example for redifferentiation are secondary xylem, wood, secondary phloem, bast, secondary cortex, cork, complementary cells. They're all examples. Xylem race, phloem race, they're all products of redifferentiation. Okay? I hope that you understood it. When a permanent tissue is forming from primary meristems, you call differentiation. When a permanent tissues are formed from secondary meristems, you call it as redifferentiation. And when you see a meristem developing from a permanent tissue, then you call that as de-differentiation. That's a very easy one to remember. Children, when we talk about a development in a cell is first is cell division, just now we discussed. After that, cell elongation. After that, cell differentiation. After that, cell maturation. Then you see the senescence. Death is not a part of development. So these are the developmental stages in a cell. Cell division, cell elongation, cell differentiation, cell maturation, and senescence. The plant organ becoming senescent because of the cells. These are all the developmental stages. Every organism in the process of development have an ability to modify itself depending on the environment or depending on the phases of growth to survive in this world. And that ability is said to be plasticity. The ability of the plant organ to modify permanently either because of the environment or because of the developmental stages is said to be plasticity. So plasticity is needed for the development. Submerged leaves of ranunculus. Ranunculus is also called buttercup. It's an emergent hydrophyte. Part of the plant is inside water, outside. Submerged leaves of ranunculus have dissected leaves for absorbing water. The aerial leaves are normal. There are two types of leaves on the same plant. Heterophilic. Environmental heterophily. This plasticity is driven by environment. Hence it is environmental heterophily. Whereas in cotton, larkspur and coriandrum, initially the leaves are with less number of lobes. Later the leaves become with large number of lobes, more number of lobes. This is in the developmental stages of this plant. Therefore, it's a wonderful example for developmental heterophily. Larkspur, cotton and coriandrum. And there are two pteridophytes which also show heterophily. One is Selaginella and Salvinia. The one more similarity between these two are, these are the two heterosporous members of pteridophytes. All right. Chalo. Now, without wasting any time, we are in the last 5 minutes of the class where we are going to discuss about the hormones. All the 4 types of hormones we are going to discuss. Auxins, gibberellins, in fact 5. Cytokinins, abscisic acid and ethylene. How to remember these hormones? Now look here what I'm doing. I'm drawing a circle. These are the side headings I'm going to use. A, where I talk about the scientists. B, where I talked about the natural hormone. Then I talk about the C, synthetic hormone. Then I talk about the D, precursor. Then E, root. F, stem. What is after F? G. All right, leaf. H, flower. Then I, fruit. J, seed. I forgot the alphabets. All right. Now look here. A is a scientist name. Then natural hormone, synthetic hormone, precursor, root, stem, leaf, flower, fruit, and seed. Still you have time to make a small column of this. All right. And just write for all the hormones, because this is the easiest way to read. By this time your teacher must have already told all this. Now A, the scientist name. What Francis Darwin, I'll start with Auxins. What Francis Darwin and Charles Darwin did. They were observing a plant, and plant started peeping out of the window suddenly. Out of nowhere, it was looking out. They got fascinated. Why it is peeping out? They observed that light is falling on one side, like this. When light is falling on one side, all the Auxins, they're sensitive to light. Because they're sensitive to light, they started moving to the shaded side. All of them started moving to the shaded side. More growth on the shaded side, because the light is falling on this side. More growth on the shaded side, less growth on the illuminated side, unequal growth, cause bending. This is called phototropism. So next time if somebody asks you which hormone induced phototropism, your straightforward answer is Auxin. Francis Darwin and Charles Darwin working on canary grass. One more scientist, F.W. Went. He removed coleoptile tip. Coleoptile, all of you know that. It is a protective covering surrounding the stem. This is the place where auxins are present. He removed the coleoptile tip. He placed it on agar block. All of you know that agar has got maximum imbibing capacity. It absorbs all the auxins. And what did he do after some time? He kept this agar block on one side. Auxins have moved only on the agar block side. More growth on this side and less growth on the opposite side. That caused the curvature. This is called Avena curvature experiment. Easy way to remember. Working on oat plant, Avena sativa, F.W. Went, very popular question. All right, with that we completed scientist. Natural hormone, indole acetic acid and indole butyric acid. Synthetic hormone, 2,4-D. You heard about in English pictures how the Vietnamese used to fight against Americans, and the America found out a way out. All right, because these people are used to stand behind the bushes and they used to arrow them down. So therefore, the Americans have used this 2,4-D in order to clear all the bushes such that the visibility increases. Now coming back to our subject, 2,4-D is a synthetic auxin which is capable of eliminating dicot weeds in a monocot crop. Weed-free lawns. I can produce weed-free lawns. That's a very important point to remember. Then precursor. Precursor for auxin is indole compounds. Indole. Root. If I take a stem tip and dip in auxins, more amount of root hairs develop from the stem tip. It is horticultural and agricultural importance. Development of roots, adventitious root at stem tip. Coming to the stem, we already discussed auxins induce phototropism. Auxins also induce apical dominance. Some of you got it out, how auxins induce apical dominance. I don't have much time, children, to discuss this. All right, but just I'll try to what is called talk how it induces apical dominance. This is the terminal bud and this is the axillary bud. More amount of auxins are transported from terminal bud to axillary bud. Then you may ask me, axillary bud should grow. That's the point. Axillary bud will not grow when auxin concentration is more. So axillary bud cease to grow. I'll tell you the reasons later. Axillary bud cease to grow when there are more auxins. So it means that the terminal bud is successful in inhibiting the growth of axillary bud such that only it can grow. This is called apical dominance. The suppression of growth in the axillary bud by the terminal bud is said to be apical dominance, and auxins are capable of inducing it. If I'm a tea farmer, well, I'm not going to feel very happy if my plant is going to grow tall. I'll be happy if there are more branches, more leaves, more profit for me. Then what I do is I'm going to carry a scissor, and whenever I see the terminal bud, I remove it. This is called decapitation. Once I remove it, the source of auxin I removed. By no way the auxins can move into the axillary bud now. My axillary bud is free of any auxin from terminal bud because there's no terminal bud. Then my axillary bud can grow now. The growth is less when there are more auxins. Growth is more when there are less auxins. There are less auxins here because there is less auxins. Why? Because I have removed the terminal bud. So in hedge making and tea plantations, decapitation is done such that there is more lateral growth. That's about the stem. Coming to leaves. Every plant, children, listen carefully. Every plant, it will flourish. It will grow properly when there is less competition for water and nutrients. Imagine if a tree contains all leaves, old, young, all. There is distribution of water and nutrients to all the leaves, and the plant cannot grow properly. Therefore, the plant uses all its common sense. The old leaves which are in the evening of their life should fall. Well, they have released enough. All right. And the young leaves which have not seen this life has to grow. This is the philosophy which is more or less nice. So therefore, what auxins do? It produces an abstracting layer, a suberized abscission layer, which is abstracting layer at the base of old leaf, such that there is no entry of water into it because an abstracting layer it in use at the base of old leaves will fall. And auxins at the same time are conscious that young leaves has to flourish. It prevents any abscission layer at the base of young, such that young leaves don't fall early. So it prevents premature leaf and also fruit fall by preventing abscission layer at the base of young, and it promotes abscission layer at the base of old. I told you the reason for that. Coming to flower, it induces flowering in pineapple. Okay, fruit, it induces seedless fruits in parthenocarpy. Before the pollen tube extends into the ovary, the fruit wall development take place. So there was no chance of any fertilization. So the fruit is there, inside that ovule is not fertilized, there is no seed. So seedless fruits, parthenocarpy, tomato. Xylem differentiation is also because of auxin. Children, I went in an order and you have understood this is the order with which you remember. In fact, you know all this. All right, that's about auxin. Then we go to gibberellins. Again, scientist, natural hormone, synthetic hormone, precursor, root, stem, leaf, flower, fruit and seed. I think I'm running out of time. So last 5 minutes we are going to put and complete this. Kurosawa, a Japanese scientist, he reported that in Japan the rice plants were growing abnormally tall and with the slightest wind currents they used to topple down. And he reported that this abnormal growth of rice plants is because of the secretions of a fungus called Gibberella fusicori, which cause Bacchanae disease or foolish seedling disease. It was later reported that not only the fungus secrete, even the higher plants have got the same chemical, therefore they rightly call them as a hormone. All right, natural gibberellins, GA1 to GA110, most common type is GA3. Synthetic gibberellins, none. Precursor, terpenes. Root, no effect. Stem, just now I told you, rapid internodal elongation of the stem. The stems of sugarcane is going to rapidly elongate in the presence of gibberellins. All right, that's an important point, which can increase the what is called profit levels of sugarcane, which increase the production of sugarcane. I'm sorry to say that. Next, we talk about leaves. No big point with regard to leaves. Coming to the flower. In cabbage and cauliflower, you have observed many times, the vegetative phase have got a rosette stem. The stem is very short, and the rosette of leaves means a wall of leaves will be there around it. Just before flowering, there is a slight increase in the vegetative shoot. Sudden increase in the vegetative shoot in the rosette plant just before flowering, that is induced by gibberellins, and this process is called bolting. Cabbage, cauliflower, beetroot, remember this. That's about flower. Coming to fruit, large size fruits in grapes and proper shape of fruit in apple. Delaying senescence in fruit trees such that in an off season if a farmer can sell his crop, he is going to get more price. So increasing the market value are the important characters regarding fruit with regard to gibberellins. Coming to the seed, look here, this is the aileron layer and this is the embryo. Gibberellins are synthesized in embryo. They move to the aileron layer of barley seeds, aileron layer. In aileron layer, gibberellins help in the synthesis of alpha amylase. This alpha amylase is going to convert the starch into maltose. Starch into maltose. So there is more amount of maltose in barley seeds. How? Because of this enzyme alpha amylase and protease. How these enzymes are synthesized? In the presence of gibberellins. So gibberellins help in the synthesis of digestive enzymes. This digestive enzymes convert starch into maltose. Maltose is the sugar which can be absorbed directly by embryo and can show aerobic respiration, can divide more faster, resulting in seed germination. In coniferous plants, gibberellins can hasten maturity and is going to help in what is called early seed production. Early seed production in coniferous plants. All right, that is also an important character for gibberellins. Now, I'm just thinking about the time also. That is the reason why I couldn't properly recollect. Same is the case for you also, children, especially on the grand test exams. Suddenly you get panicked, all right, whenever you see the watch. All right, please don't do that, right? Just be very cool. You'll have enough time. The composure, whoever has going to hold their nerve and are more composed is going to win this game. Now it is all the psychological aspects which play the role now, because everybody knows everything now. So whoever can retain all the points with a lot of calmness and composure will be standing ahead. Am I doing it now? I think so. All right. That's about gibberellins. Now we go to the next hormone, cytokinins. All right. Skoog and Miller. All right. They have isolated a herring's fish. From there, they have isolated a sperm cell. From sperm cell, they have isolated the DNA. From the DNA, they autoclaved the DNA, heated the DNA, and isolated 6-phosphorylaminopurine, a nitrogen base, which is capable of causing rapid cell division, cytokinesis. Therefore, they call it as kinetin. Later, it was Skoog and other co-workers isolated a natural cytokinin, because kinetin is neither natural nor synthetic, autoclaved product. So how can it be natural? All right. So it was Skoog and other co-workers isolated a natural cytokinin from endosperm of maize and coconut. Maize, zea maize. So therefore they call it as zeatin. All right. That's the scientist names. It caused rapid cell division in tobacco pit cells. So that's about the two scientists. Then we talk about natural cytokinin. It's an adenine, isopentenyl adenine. Synthetic cytokinin, benzyl aminopurine, not in your book. You can successfully forget, not needed. Precursor. Precursor is nitrogen bases, purines. All right. Then we talk about root and stem. Now I'm going to talk about root and stem put together. Root and stem. More auxins, less cytokinins from callus. Callus means undifferentiated mass of tissue in tissue culture. It helps in the development of root from callus. More auxins, less cytokinins. This is called rhizogenesis / morphogenesis, where auxins and cytokinins are joining hands, synergistic effect. Same way, more cytokinins, less auxins will help in the development of shoot. All right, development of shoot. This is called collagenesis / morphogenesis. So in tissue culture, we have to use these two hormones, children, for the development of callus into a plantlet, auxins and cytokinins. Both of them should be there. All right, that's about root and stem. Coming to leaves, flowers and fruits. One point I'll tell you, all three common. Nutrient mobilization. More amount of nutrients will move into the leaves, flowers and fruits because of cytokinins, because cytokinins induce rapid cell division, all right, and thereby more amount of xylem and phloem is produced, more amount of water and minerals enter into leaves, flowers and fruits. When there is more water in leaves, flowers and fruits, the aging process is delayed. You cannot prevent, either in plant or in you. Aging cannot be prevented. Aging can be delayed. Senescence is delayed. And the delay in senescence is because of nutrient mobilization, which is brought by cytokinins in leaves, flowers and fruits. They increase the shelf life period. They increase the vase life period in flowers. All this is because of delay in senescence. That's it. That's about cytokinins. Then comes ethylene. It's a widely used hormone in agriculture and horticulture. Cousins for the first time reported that ripe oranges are going to release ethylene, which can help in the ripening of unripe bananas. All right, then all of you know that ethylene is a gaseous hormone. Ethyphone is an ethylene releasing chemical. Now, going in an order. All right, natural cousin, scientist name, over. Natural ethylene. Precursor for ethylene is methionine actually, but it's not in your book. All right, just leave it. Ethyphone is an ethylene releasing chemical. That point is there. All right. Then next important one is root system. It helps in the development of more number of root hairs. Increase the surface area of absorption, very important. Coming to the stem system, it helps in the increase of the internodal length of underwater rice plants and also the petioles. such that they can come out of water, because more water is added in rice fields during seed germination and seedling production. Increase in the internodal length of underwater rice plants. Remember, ethylene. Then after stem, leaves. It induces abscission layer at the base of young. We already discussed auxins prevent abscission layer at the base of young. Ethylene has more inhibitory effects than promotory. Ethylene induces abscission layer at the base of young leaves. Number of female flowers in cucumber. At the induce abscission layer at the base of flowers in cotton, cherry and walnut. This is called thinning of cotton. Thinning and the formation of an abscission, obstructing layer. Not only at the base of leaves, young leaves, but also at the base of flowers in cherry, cotton and walnut. Remember that is called thinning of cotton, cherry and walnut. That is run by ethylene. Then coming to fruit, you study in organic chemistry also. Ethylene is responsible for fruit ripening. It helps in fruit ripening. It helps in the synthesis of an enzyme pectinase. That pectinase dissolves middle lamellar. When all the cells of the fruit wall get separated, the fruit wall becomes soft. Simple. All right. Then increase in rate of respiration at the time of ripening, respiratory climatic. Ethylene has a role to play. Then after that, seed. It helps in apical hook formation, the swelling of the dicot seedling and horizontal growth. All these three put together is said to be triple response growth. Triple response growth is also a bioacid test for ethylene. Avena curvature test is a bioacid test for auxin. Dwarf plant growing tall, dwarf mutant variety test is a bioacid test for gibberellin. Tobacco pith culture test is a bioacid test for cytokinin. Triple response growth, thinning of cotton, cherry and walnut are the bioacid test for ethylene. Okay? And ethylene also helps in seed germination in peanuts and helping in the sprouting of potato tubers. I told you in an order, just like your teacher. So therefore, there is no big problem in understanding this. Then we talk about abscisic acid. Is a growth inhibitor? Yes. You may ask me why the plant is going to produce a growth inhibitor. All right, knowingly, because at sometimes the growth should be inhibited for the welfare of the plant. Imagine outside conditions are not favorable, very hot. The growth should be stopped, and for that abscisic acid is needed. Outside conditions are very hot. The plant doesn't want to lose water. The stomata should close. Abscisic acid does the job of permanent closure of stomata, if not permanent, closure of stomata, and thereby the water can be conserved. Water is not allowed to move out. So does abscisic acid a growth inhibitory hormone? Answer is yes. Does it cause a lot of stress by causing growth inhibiting activities? No. It relieves stress by closure of stomata, by inducing dormancy. It allows the plant to survive the stressful conditions. So it is not going to cause stress. It's going to relieve the plant from stress. So the two important points, children, about abscisic acid. Precursor for abscisic acid is carotenoids. You remember that? Dormin, abscissin 2, all right. They're all abscissin. They're all the chemical compounds which are more or less similar. All right. Later, all of them put together is called abscisic acid. Only 2 points about abscisic acid I want you to remember. One, it's a growth inhibiting hormone, unlike ethylene, which has got both inhibitory and promotory, more inhibitory than promotory. Here only inhibitory, abscisic acid. 2, the precursor of abscisic acid is carotenoids. Remember, a pigment is a precursor of a hormone. What is a pigment? What is a hormone? Carotenoid is a pigment, hormone is abscisic acid. Third important point, abscisic acid causes closure of stomata, is an anti-transpirant, very popular question. 4, it helps in seed dormancy. Induces seed dormancy. What about gibberellic acid? It allows seed germination in barley. That allows seed germination. This allows seed dormancy. Antagonistic, opposite effects. That's a very popular question. Gibberellic acid and abscisic acid are antagonistic with regard to many activities. So seed dormancy, seed maturation, all of them are the effects of abscisic acid. That completes this chapter totally. I made an attempt to include every line which is there in the textbook, but again I can tell you that maybe I may forgot 1 or 2 lines here and there, but this is a tiny attempt, children. Please read the textbook thoroughly. All the best, and we'll meet tomorrow with one more chapter.